Hi Planet Earth students, this video is going to provide an introduction to our next unit on the Cambrian explosion. And the Cambrian explosion really starts with a scientist's discovery of a very odd fossil bed called the Burgess Shale. And so I'm going to go into that discovery and what has happened over the past 100 years to advance that research forward. And we'll end up talking about the Cambrian explosion as a very odd event in the history of biology on Earth. So the Burgess Shale, this fossil formation central to the Cambrian explosion, was initially discovered by a team led by Charles Walcott, specifically Charles Doolittle Walcott. Very important you know that he had a very interesting middle name. But this guy was a really eminent scientist in the early part of the 20th century. He was a professor at Harvard. He directed the Smithsonian for a while. Very important guy, and he took a trip in the early part of the 20th century, 1909 specifically, to a spot, I'll show you it on a map here in a second, called Yoho National Park. This park is on the western coast of Canada in British Columbia. And what he discovered in this national park was a very odd fossil bed. He found these fossils in a formation called the Burgess Shale, and we've talked about shale rock before in Planet Earth, pretty similar stuff. The reason why the fossil bed was so odd to him and to science is, firstly, the stuff he was finding, he had never seen anything like these organisms that were fossilized before. He had a very difficult time kind of connecting the look of these organisms to anything that existed in the current day. So they looked very weird and seemed completely new to science at that point. And the second thing is, normally, fossils represent hard parts of the organism that have been preserved, and hard parts are pretty easy to preserve, and so that's what scientists see after millions and millions of years. But these fossils in particular had very good preservation of soft body tissue, so he was able to see both the hard parts of these organisms and a lot of their soft tissue, which had been preserved because of the strange environment in which this shale had been deposited. So Walcott ends up dedicating a large part of his career and his laboratory effort to this fossil bed. He returns well over 10 times to this area to collect specimens, and he ends up amassing quite a few fossils. I've written here over 65,000 fossils in around 10 years by 1924. So he investigates this fossil bed quite thoroughly. And I'll show you some examples of the things that he discovered here. So here's a map of where this location is. It's on the border between two Canadian provinces, British Columbia and Alberta, just west of Calgary, Alberta. Yoho National Park, where this Burgess Shale formation is. And if you've been to this area of Canada, it's very beautiful. Definitely a spot that you should visit in your lifetime. Some of you might have been to Jasper National Park, which is a famous national park up here. You might have also been to Banff National Park, which is located right in there. So there's a lot of really cool natural areas around here, and if you go up there, you can definitely stop by Yoho National Park, although they definitely restrict access to the Burgess Shale specifically. I've tried to get there myself, and it's a bit tricky. But lots of very interesting fossils were found here. So here are some example images of Burgess Shale fossils, and the first thing I guess I want you to notice is how weird these things look. I mean, you can look at this organism up here. What exactly is that? Very odd looking. Look at the hard body preservation in this organism here. Amazing. You can actually see the individual appendages of this organism in the fossil. But more importantly, as I mentioned before, a lot of these fossils had very good soft body preservation. So take, for example, this organism down here. That looks like all soft tissue, and you can still see a lot of detail in the organism, even what look like very fine pieces of soft tissue coming off of the side of the organism. So really cool fossils. Here are some other examples of Burgess Shale fossils, just to give you an idea of the diversity that was present in this fossil bed. Very cool stuff. This one here they called hallucigenia, specifically because they thought they were hallucinating when they were looking at it. It was so odd looking. So again, some more examples. And here's some more examples. Still different organisms present in the Burgess Shale fossil bed. So lots and lots of biodiversity in this fossil bed. Most of the stuff is stuff that was very difficult for Walcott to assign to any phylogenetic category, and really, really good fossil preservation. So I want to focus here on how Walcott interpreted these fossils. And let's first think about what Walcott thought about the phylogenetic tree and how evolution worked at the time. He was aware of Darwin. He was a scientist at the time that subscribed to the theory of evolution. So think about what his view of evolution would have been. And it might be similar to what your view of evolution is now. Walcott's view of evolution, to simplify, is basically a slowly branching phylogenetic tree. So you start with one beginning of life early progenitor, and you get slow diversification via natural selection and adaptation until you get a present level of biodiversity. So what he did is, based on this view, he started trying to systematically classify these odd specimens he was getting from the Burgess Shale into existing phyla that he knew of at the time. So he said, okay, I'm looking at this weird Burgess Shale fossil. I'm going to give it a phylogenetic classification based on what I think it looks most similar to that's present and living today. And so in a small scale activity, what we're going to have you do is attempt to do what Walcott did. Here are drawings, which are the product of quite a bit of research in the fossils, drawings of 6 of the most famous Burgess Shale fossils: Sancticaris, Wawaxia, Hallucigenia, Morella, Deinonychus, and Picea. 6 of the most famous Burgess Shale fossils. And what we're going to have you try to do is classify these based on currently existing phyla of organisms. Here is the activity sheet that you're given. So what you can do with this activity sheet is go ahead and write the name of those 6 organisms over here on the organism name side. And of course, write your name up here in the top corner. And what you're going to want to do is, as you make your phylum classifications, you are going to write what you think that particular Burgess Shale organism is in this column here for your phylum classification. And what I also want you to write is a brief rationale for why you classified that particular organism in the way you did. So, for instance, you might classify a certain organism as an arthropod. Okay, why did you do that? What were the aspects of its specific body plan that led you to make that classification? And you'll record that brief rationale for each organism in this column here. Go ahead for now and leave these other columns about different scientists' interpretations blank. As we review this activity, you're going to be filling these out and coming to the realization that different scientists have interpreted these fossils in very different ways. And we're giving you some stuff to help you in making these classifications. You're going to want to use this handout packet called Brief Survey of Organisms. And what this handout packet includes are all of the different currently existing phyla, along with images of organisms that are in those phyla. Each image has with it a size comparison, so you can kind of get an idea of how large the size is in comparison to the image on the sheet of paper. And each phylum also has with it a description of the different types of body plan characteristics and other things that identify or help you identify that particular phylum, which will help you in completing the information required for the rationale column. So this is a review on this slide. You can pause it here and kind of look over what you need to do. So you've been, again, provided with sketches of 6 of the important Burgess Shale fossils, and you're going to assign each one of these organisms, give it your best effort to the phylum you think most appropriate, and your reference for doing that is this brief survey of organisms handout packet. Use major body plan characteristics to do that, and then provide a rationale. And make it a simple rationale, but not a completely blank rationale. I want you to use body plan characteristics and not just say, it looks like a snake, so it's, a snake. Maybe make it a little bit more specific based on some of those definitions and body plan characteristics listed in the brief survey of organisms. And leave the interpretations of the other scientists and the current interpretations part blank. This will eventually be an 8-point grade when you complete it and turn it in. So give it your best shot. So at this point, you can pause this or stop it, and I actually want you to spend at least 15 minutes going through and trying to assign the different Burgess Shale fossils to phyla. Okay, so at this point, hopefully you have the first 3 columns of that worksheet completed. And you've given it your best shot and tried to identify each one of those 6 important Burgess Shale fossils, try to give it a specific phylum categorization, and written a brief rationale for why you think so. Again, the rest of the sheet's blank, and that's what we're going to be completing throughout the rest of this review. So one thing I wanted to ask you at this point is I intentionally didn't give you certain information. And so I want you to think, other than looking at these specific sketches that I gave you, what other data would you need or would you have liked to have in order to make your guesses better? So normally when we have this discussion in class, people instantly offer up that they would like to know how large these fossils are, which is a solid point. And I intentionally didn't give you that information, but I'm going to give it to you now. So here's a comparative, a size comparison of 3 of those organisms, Wilwaxia, Pichia, and Hallucigenia, and the other ones that I gave you are very similar in size. And you can see here the outline of a human. There's a leg. So this big blue thing here is a human, so you can get an idea of the comparative size. But very, very small, and you can see up here this is 10 centimeters in size, not actually on your screen right now, but that's the size comparison. So very, very tiny organisms. And that's important to know, and that might have helped you quite a bit in making your phylum assignments. So remember that these organisms are very small. A second piece of information that students typically would like to know to make their guesses better is what did this environment look like where they were living during the time in which they were deposited. And that would be the Cambrian period. And this diagram here shows you what the Cambrian period looked like during the later part of the Cambrian, around 514 million years ago. And what you can see obviously the continental margins and stuff like that. There was a larger continent called Gondwana at that time, which we've briefly alluded to in that Mass Extinctions Review series. But you might have guessed that these were marine organisms, and you would have been right. The Burgess Shale would have been a very coastal type of environment. That shale was being deposited on a seafloor, much like central Texas during the Cretaceous period. And you can see it here, Burgess Shale, right off the coast of the continent, Laurentia. So it's important to note that the Burgess Shale fossils were marine organisms, and they were deposited accordingly on that seafloor in the Burgess Shale Formation. So what we're going to go through and review now is how Charles Walcott interpreted these fossils, and I want you to compare his interpretation to yours. So on the left-hand panel here, this is what you were looking at, a drawing of this organism. This is an example of what Walcott would have had, a fossil, so a lot of the same information. Look at what you interpreted this organism as, and look at why you interpret it to be that particular thing. And here is what Walcott thought. So Walcott thought this was an arthropod, and at the time, arthropod was a phylum, Now it's a superphylum that contains the phyla Chelicerata, Crustacea, and Mandibulata. So if you put any of those phyla, you would be agreeing with Walcott here. His rationale was that it looks like it has an exoskeleton, it looks like it has body segments and jointed appendages. And this sort of superphylum would include insects, crustaceans, arachnids of today, and people doing arthropod projects in planet Earth are very acquainted with it. So I want you to write arthropoda next to Morella in terms of Walcott's interpretation. And then, put a check mark or an X mark there based on whether or not your definition agrees with Walcott's. And that's not for a grade. I just want you to be kind of keeping track of this because that's what makes this interesting, at least to me. Let's move on to the next one. So, Sancticaris. Left-hand panel is what you had. Here's an example of a Sancticaris fossil that Walcott looked at, currently in the Smithsonian. Take a look at what your interpretation of this organism was and why. And here's what Walcott thought. Welp, Walcott actually didn't know. So he thought, I don't know what this is. And so he spent a lot of time trying to assign it a category, but in the end, he reached no definitive conclusion, and so his notebooks express that conclusion. So you can write in the blank next to, for Sancticaris, Walcott interpretation unknown, and I doubt anyone put that unless you kind of bowed out on this assignment. So you probably took an educated guess on this, and who knows, you might be right. We'll see later, but definitely put unknown for Walcott's interpretation of Sancticaris. Okay, Pikaia. This is going to become one of the more interesting ones that has been reevaluated quite a bit over the past 100 years. Take a look at what you thought this was, and why you thought it to be that particular thing. You might be thinking at this point, well, which side of this organism is the head and which side is the tail, or does it even have a head or the tail? So a lot of people interpret this as sort of the front or head of the organism. A lot of people interpret this as the front or head of the organism, and that might make a little bit of a difference in terms of how you're interpreting what the organism is. And then look at what Walcott had. I mean, you might think that that's kind of a bummer of a fossil, but it's incredibly articulated, especially when it comes to the soft parts of the organism. Let's take a look at what you put, and here's what Walcott thought. So Dr. Walcott assigned this to a phylum called Annelida, which consists of ringed or segmented worms. The reason why he did that is because he believed that this organism moved. He noticed that it was bilaterally symmetrical, so you can kind of draw a line down the center of this organism, and its body plan mirrors itself. And it looked to him like it had a sort of a segmented body, so he would call these things little segments. And you can even see those little segments over here. That's what he was calling a segmented body plan. And today, that phylum includes ragworms, earthworms, leeches, that type of thing. So put a little check mark or an X mark. Some of you might have identified this as Annelida. At this point, who knows if you're actually right, but we're just looking at what Walcott thought. But it's cool to see if you agree or disagree with Walcott. So he thought this was an Annelida, and you can evaluate that as compared to what you thought. Let's move on to the next one. Wawaxia, definitely the oddest named one of the 6 that we're dealing with, unless you count hallucigenia, which is kind of weird too. So left-hand column is what I gave you. Walcott was left with fossils that are a little bit more difficult to figure out. And again, this thing that you were looking at is a product of about 100 years worth of work and kind of reconstructing what this thing actually looked like. So Walcott, remember, didn't have nearly as much with this particular organism. But you might not like his interpretation. Compare it to yours. Walcott also assigned this to the phylum Annelida. So he thought that this was also a ringed, segmented worm thing. And he thought so for the same reasons. Now, based on the drawing that I gave you, it's pretty clear that maybe it's not that. And so we'll go through some reevaluations of Wawaxia throughout the rest of this presentation. But that's what he thought based on the limited amount of information that he had. So go ahead and write down Annelida under Walcott's interpretation of this Wiwaxia fossil. And you can compare it to yours. Maybe some of you thought this was a ring segmented worm. Typically in classes, not too many people do, but definitely compare it to what you thought. Dynamicius. So again, look at what Walcott was dealing with. Definitely not as much as you had. I think an interesting question comes up when I do this in class that some people, usually a group of people, think that this is a plant, and then a group of people think that it's not a plant. So I guess one question is, do you think this is a plant? You're probably not going to be very impressed with what Walcott thought, but take a look at what you thought in comparison. Walcott was not able to reach a conclusion on this particular fossil. He did not reach a definitive narrative as to what phylum to classify it as. So he said he did not know. So write down unknown for Dynamicius next to Walcott interpretation. But pay attention to what you thought that this was. Last but not least, Hallucigenia, my personal favorite. So a very odd-looking organism. You might have had some hints about this organism based on your readings about the Cambrian explosion, or your prior knowledge coming in. This is the most famous of the Burgess Shale fossils. So take a look at what you interpreted this as. And for Walcott, you're once again going to write down Annelida, ringed segmented worm. He thought that a lot of the Burgess Shale fossils were these worm-type things, based on him believing that they moved, his observance that they were symmetrical bilaterally in terms of body plan, and his observation of distinct body segments. So he thought that this was an evolutionary ancestor of ragworms, earthworms, leeches. So write that down for the Walcott interpretation and compare it to yours. Maybe some of you thought that this was a ring segmented worm as well. And we'll see how the narrative continues. So let's talk about what happened after Walcott. And I want you to pay attention to these dates up here, 1960 to 1970. Think about when Walcott was doing his work in the early part of the 20th century. So it took a very, very long time for scientists to come back and look at this work again. And honestly, the Burgess Shale fossils were kind of put in drawers at the Smithsonian and for the most part forgot about for over 20 years. But in the 60s and 70s, there were three prominent scientists, amongst others, that served to reevaluate Walcott's findings in light of new technology. And those were Harry Whittington, Derek Briggs, and Simon Conway Morris. A couple other things happened after Walcott that are equally important. Three new really important fossil sites were discovered that had fossils similar to those of the Burgess Shale in China and Australia. And a third thing, lots more information was learned about what life was like during the Precambrian. The immense amount of time, the billions and billions of years prior to the Cambrian period, where there was life, but life was very simple. And they discovered fossil evidence of this life that helped scientists learn more about what was going on during that time period. So what do we currently know about life during the Precambrian period? Well, we know that no organisms were more complex than little squishy things. And what we mean by little squishy life means bacteria, some multicelled algae, some single-celled, very simple plankton. And because we're describing this as little squishy life, kind of weird, but I've heard that multiple occasions, there's going to be limited fossil evidence of this. And so I guess I want to share with you, maybe you already know, what the fossil evidence is of this life that was little and squishy, and why particularly that fossil evidence suggests a very slow rate of evolution. Not much is changing over these billions and billions of years of the Precambrian period. What we know of the Precambrian period comes primarily from specific fossils called stromatolites. Here is a picture of modern stromatolites, and here are some of the Precambrian fossil stromatolites that have been discovered. And this is a particular type of organism that actually served to trap, bind, and cement sediment. And it did that as part of its metabolism. So these organisms would slowly, over the course of their life, trap sediment and deposit a layer. Trap sediment, deposit a layer. Trap sediment, deposit a layer. And they did that for a very, very long period of time. And so what you see in the fossil record as evidence of these organisms' life is not the organisms themselves, but what they left behind, those layered sediments. And so you can see here, and you have to have a very trained eye to actually make anything of this, and I don't have that trained eye, so it just looks like a sort of a squishy layered mess to me. But that is evidence right there of living material that served to produce that. It's much easier to interpret that evidence when you have a living record of these things doing this exact same thing. So there are organisms living on the surface of these little boulder-looking things that are serving to actually continually create the boulder through trapping and cementing sediment. So what this fossil evidence suggests, because stromatolites are the only major form of Precambrian fossil evidence we have yet discovered, it suggests that yes, Precambrian life, you remember life originated around 3.8 billion years ago. For billions and billions of years after that, life is little and squishy, no hard parts. And what we also know, based on looking at stromatolites over time from different time periods, is that life was not evolving very much, a very, very slow rate of evolution. And that makes the Burgess Shale fossils and the fossils from these other beds that we're going to talk about a lot more important, because these fossil beds show us the first evidence of the development of hard parts, and also show us a very, very accelerated rate of evolutionary process happening near the beginning of the Cambrian period. So I mentioned that another important thing that happened after Walcott was the discovery of a lot of other fossil beds around the world that had fossils very similar to the Burgess Shale. Three that we're going to draw specific attention to. Here's the Burgess Shale up here again. There's Chengjiang fossil bed, which is in what's now China, and the Emu Bay fossil bed, which is off the coast of Australia. And those are three of the more famous ones that we're going to be discussing. But there are quite a few more locations where fossils similar to those found at the Burgess Shale have been found in the world, and those are all labeled on this map with yellow dots. But this is a map of the sort of modern-day landscape of the world. Let's look at what this environment would have looked like during the Cambrian period. So here's what the landscape looked like during the Cambrian period. This is specifically from the late Cambrian period. And again, to remind you, this is the Burgess Shale right off the coast of the Laurentia continent. And if you look at where the Changjiang fossils were then, they were in a similar location, right off of a coast. Emu Bay as well, right off of a coast. And it's important to note that because Burgess Shale style fossils were discovered around the world, what this means is this weird development of strange fossils in the record of life on Earth wasn't an event that was anomalous. It wasn't an event that was localized to a specific area in British Columbia and just happened there. No, it means that it was a global event that really redefined the entire landscape of evolution. Because remember, what's happening prior for billions of years is not much, little squishy life, not doing much, a very slow rate of evolution. And then it appears from the existence of these fossil beds around the world that something crazy starts happening evolutionarily in a very short period of time. And that period of time was around 550 million years ago, near the beginning of the Cambrian period. Another cool fossil bed that was discovered was also in Australia, and it's the Ediacara Hills fossils. Now you can see, remember the time that I just mentioned was 550 million years ago, around the beginning of the Cambrian period, so specifically 542 million years ago, but you can put the time 550 in your mind. And the Burgess Shale fossils, for the most part, date to around 505 million years ago. Emu Bay off Australia date to around 520 million years ago. Changjiang fossils date to around 525 million years ago. And what these fossils from the Ediacaran Hills allow scientists to do is realize the transition. So we kind of have a before and after picture. And even 600 million years ago, life is starting to do really weird things. So what we'll do now is look at some examples of fossils from the Ediacaran Hills, Changjiang, and Emu Bay, and you can compare and contrast those to those found at the Burgess Shale to get an idea of how life is changing very quickly during the late Precambrian and early Cambrian. Here are some photos of fossils from the Ediacaran Hills. They date to around 600 million years ago, so before the beginning of the Cambrian period. Probably the most famous of these is the Dickinsonia fossil. You might have seen something that looks like this before. And you might notice that these definitely look like these organisms had hard parts, at least it looks like to me. But they're not as ornate and complicated looking as those from the Burgess Shale. And so you can kind of get an idea of the transition here. This is 600 million years ago. The Burgess Shale fossils date to around 500 million years ago. So 100 million years of evolution takes you from this point to the Burgess Shale. But remember, if you compare this to stromatolites, obviously these organisms are wildly different. So the evolutionary process is starting to radically change as far back as 600, even further back million years ago. Here are some photos of fossils from the Changjiang fossil bed that date to around 525 million years ago. And you can compare them to Burgess Shale fossils, again, around 500, 505 million years ago. And these are starting to look a lot similar. So the previous ones from Ediacaran Hills, 600 million years ago, Changjiang fossils are Cambrian fossils, early Cambrian fossils. And you can start to see the development of more complicated body plans. Here are some other examples of fossils from the Changjiang fossil bed, which is currently China. And you can see things that look quite similar to stuff that Walcott found in the Burgess Shale. Take a look at this. I mean, I think that this looks fairly similar to that Hallucigenia fossil without the spiky things. And so the similarities are very important to note. Again, this was a process that was happening around the world at a similar time. Here are some photos of fossils from the Emu Bay bed, and you can notice similarities as well. I mean, there's a very similar thing to the thing I circled in the previous slide that was found in China. These date to around 520 million years ago. This again showing that similar process was happening globally. So the next major development in this story is Stephen Jay Gould. Gould is a scientist, but also a science writer. And so he wrote a book called Wonderful Life, where he wrote about the Burgess Shale fossils, the history of Walcott, and also made some conclusions of his own regarding these fossils. And this was in 1989. And so what the book did, it was a very popular bit of science writing, and it's a good thing to read. It's well written. But what this did is, for the general public, repopularize these Burgess Shale fossils. And he provided a summary of the reevaluations of scientists like Conway Morris, Derek Sprigg. But in the book, he also draws some very controversial conclusions about the identities, the phylum classifications of these organisms. And so we're going to review those. So if you look back at your activity sheet, you should have for the Morella fossil Walcott's interpretation as an arthropod. And let's look at how Gould interpreted this fossil in that book, Wonderful Life. So Gould actually agrees with Walcott. He also categorizes this as an arthropod. So what I want you to do is on your sheet there's a column for Gould interpretation, and this was in 1989. Write down Gould's interpretation of Morella as an arthropod. And in the end, arthropod is a superphylum that contains a lot of the phylum that are known as separate phylum today. So if you put arthropod or something that is a subcategorization today of an arthropod, then you would also be agreeing with Gould. On to Sancticaris. So Walcott did not know what this was. How does Gould interpret it? So Gould interprets this also as an arthropod. And Arthropod is a pretty common interpretation when I do this work with class. A lot of people think that this is an arthropod, and you'd be agreeing with Gould. He thought that this was an arthropod based on a lot of those body plan characteristics, and joints and appendages that look like they have joints and things like that. So go ahead and write down for Gould interpretation of Sancticaris, arthropod. Okay, so how did Gould interpret Pikaia? So this was super controversial, but Gould interpreted Pikaia as chordata, which is a superphylum containing Urochordata in your packet, Cephalochordata in your packet, Craniata, and others in your packet. So the reason why this was controversial is Gould was interpreting this not as a worm like Walcott. Remember, Walcott interpreted this as Annelida, a ringed segmented worm. Gould interpreted this central thing here as a notochord, which would be an ancestral spinal cord. He also found in this fossil what he thought were pharyngeal slits. And what pharyngeal slits are filter-feeding organs that are found in vertebrates. And you can kind of see some evidence of those on that drawing there. There are these repeated openings that exist right sort of below the mouth in the body plan, and apparently they allow for water to move into the mouth and out of the slits. He also defined in this organism what he thought was an endostyle, which is an organ that produces mucus, and it's thought that that mucus is secreted to specifically trap food particles and assist in this filter feeding. So you can't really see that on the drawing I gave you, which is unfortunate, but you can vaguely see it in this fossil photo, be like right there. And lastly, he defined in this organism a post-anal tail, which is a characteristic of vertebrates, a tail that exists past the anal opening. So it would be that thing right there. So why was Gould's conclusion that Pikaia was a chordate so controversial? Well, what that suggests is that this organism that appears to have evolved during this Cambrian explosion period, in the early Cambrian period, and is very present in the Burgess Shale fossil bed, is the earliest direct evolutionary ancestor of vertebrates, which would be fish, amphibians, reptiles, birds, and humans. And so this was very controversial. And that was one of the more startling conclusions in his book. So I want you to write down Gould's interpretation of Pikaia as a chordata. So on to more controversial conclusions from Gould. So Walcott thought that Wiwaxia was a ringed segmented worm, which you might have disagreed with. It's hard to see why he thought that. Let's check out what Gould thought. So Gould thought that this was a new phylum. And what he meant by that was a phylum that existed at the time of that evolution in the Cambrian period, but that no longer exists today. So basically, if you adopt Gould's interpretation of this fossil, the assignment I gave you would be impossible, because this organism cannot be categorized in any phyla present in that packet that I gave you, because this phylum no longer exists. And so this was controversial because it was a reinterpretation of evolution, and I'll show you kind of why that is here in a second. So for Gould's interpretation for Wiwaxia, go ahead and write down new phylum. On to Deinonychus. Walcott did not know what this was. Gould interpreted this similarly to the way he interpreted Wiwaxia. Namely, he defined it as a new phylum. He thought that this was a phylum that existed in the Cambrian period that no longer exists today. So write that down for Gould's interpretation of Deinonychus, new phylum. And lastly, my personal favorite, Hallucigenia. Walcott thought this was a ringed segmented worm. Gould also thought that this was a new phylum, believing that he could not assign it to a phylum today because the phylum of this organism no longer exists today. So, why were these new phylum identifications controversial specifically? Well, they were controversial for a variety of reasons. One reason that they were controversial is they established the existence of this this thing that we're going to henceforth be calling the Cambrian explosion. In that life wasn't doing much for billions and billions of years after 3.8 billion years ago when life evolves. Evolution is very slow, little squishy life, and then at this period around 550 million years ago, there's a geologically sudden explosion and diversification of life. And that's what we're calling the Cambrian explosion. So that was controversial because that went against, remember what Walcott's evolutionary tree of life looked like, slow, gradual diversification. This would not be slow, gradual diversification. This would be a very, very quick evolution of most of the phyla. But further, what made this conclusion controversial is because of all those new phylum definitions he was offering. What he suggested is that after this Cambrian explosion, most of the phyla that evolved during the Cambrian explosion went extinct, and it was only a certain few that survived to the present to constitute our current biodiversity. So you can see here that most of these phyla that are evolving after the Cambrian explosion go extinct in the evolutionary record. But then a couple serve to constitute all of the phylogenetic biodiversity that we have today. So that was controversial. And then the last and possibly most controversial thing was that he offered that the main factor that decided whether or not a phyla would survive or not was chance, complete happenstance. And this went entirely against Darwin, which is not based on chance. It's based on evolutionary fitness. So this was a circumstance that he was describing that kind of violated the principles of evolution in a sense. So it's important to know at this point that all of Gould's conclusions were not right. At least current scientists do not agree with them. And we'll evaluate that more later. But firstly, one thing that he did get right, at least in terms of what current scientists think, is the existence of this Cambrian explosion of life. And so we're going to talk a little bit more about that now. And then we'll get on to evaluating what scientists currently believe the Burgess Shale fossils are. So it might help you at this point to get out your geologic timescale handout, really the sort of the timeline for this course, to recognize where we are specifically right now in that timeline. And what we're looking at, this Cambrian explosion event, is right here, around 545 million years ago. You can think 550 million years ago, around the beginning of the Cambrian period. And this next portion is going to serve as a review of an article that you've read called When Life Exploded, a Time magazine article that summarizes the Cambrian explosion as an event. And if you haven't read that, definitely read it. It's a very good summary of a lot of this material. So here we are again on our course timeline. We've talked at this point about the geobiology of the present. We've talked quite a bit about the geobiology of mass extinctions. And in this is specifically, we're going to talk about physical things that served to create an environment in the world that allowed for this very weird Cambrian explosion event to happen. So what specifically was the Cambrian explosion? It started around 543 million years ago at the beginning of the Cambrian period. If you're trying to remember when it started, you can think 550 million years ago, and that's good enough. But what it was a rapid explosion of life. And what I mean by rapid is very geologically rapid. We're talking within around 10 million years of time. And that is extremely geologically rapid. Remember that for billions and billions of years prior, not much was happening evolutionarily. And during this 10 million year period, all animal phyla but one, and that one is Bryozoa, which is a phylum of aquatic invertebrates, all those except for one appeared during that brief 10 million year period. And so we know based on this that the rate of evolution during this period was very, very accelerated. This was a very fast diversification of organisms. And we also know, because fossil beds have been discovered around the world that illustrate this, that this event occurred at relatively the same time globally. And to remind you of four of our main sources of fossil evidence for that, we have evidence from the Ediacaran Hills in Australia that dates to before this occurred, but still you can see the evolution of hard parts and things like that. And then we have three fossil beds that are kind of post-Cambrian explosion that show this rapid diversification of phyla. So let's finish your chart off now and talk about what science currently thinks about the phylum identifications of the Burgess Shale fossils. So for Morella, look at what you thought this was, and then remember that Walcott thought this was an arthropod, Gould thought this was an arthropod. The current interpretation of this fossil is that it is an arthropod. So a lot of you probably got this right if you put anything that falls under that arthropod superphylum. And Walcott notably got it right, as did Gould. So, well, I say got it right, we might have it wrong now, but our best guess currently about this fossil is that Morella is a very early, one of the earliest arthropods. So go ahead and write arthropod or arthropoda under the current interpretation column for Morella. Let's look at Sancticaris. So Walcott thought that this, he didn't know what this was, didn't make an interpretation formally of this fossil. Gould thought that this was an arthropod. The current interpretation agrees with Gould in saying that this was an arthropod. So if you put arthropod for this, or anything that's categorized, remember arthropod was a superphylum, so anything that's under that, mandibulata, chelicerata, crustacea, those types of things, then your interpretation would be agreeing with what science currently thinks. So go ahead and write arthropoda down for current interpretation of Sancticaris. Okay, Pikaia. Walcott thought ringed segmented worm, Annelida. Gould controversially thought that this was one of the earliest chordates. And maybe shockingly to you, that's what our current interpretation is. So if you put, remember chordata is a superphylum, if you put Urochordata, Cephalochordata, Craniata, those types of things, then you would be agreeing with our current interpretation of Pikaia, is that it is one of the earliest chordates. And so that might be very odd to you, looking at, I guess, our most ancient, at least direct evolutionary ancestor. But that's what our current interpretation is. So write down chordata under current interpretation for Pikaia. Let's move on to Wawaxia. Walcott thought that this organism was a ringed segmented worm. Gould disagreed, and remember he thought that this organism was a new phylum that existed in the Cambrian period that no longer exists today. Our current interpretation of Wawaxia is that it is a very early mollusk, and mollusca is a phylum that is really large and diverse. It includes snails, slugs, mussels, octopi of today, which are very diverse species. But look at your interpretation initially. You might have gotten this right. If you put mollusca, then congratulations. But that's what we currently think this is. And it's important to draw this distinction here between what Gould thought and what the current interpretation is that no, current scientists are disagreeing with Gould in that Wawaxia was not a new phylum that no longer exists. They're assigning it to a phylum that does exist. And so that's going to revamp what our idea of the evolutionary tree of life looks like. And I'll show you that here in a second. So if you haven't done so already, go ahead and write mollusca for current interpretation of Wawaxia. Deinonychus, confusing one for Walcott. He did not make an interpretation of this fossil. Gould, controversially again, thought that this was a phylum that existed in the Cambrian but no longer exists today. And, well, what do you know? We've gone back to kind of the beginning. The current interpretation is that we have no idea what this is, which is kind of funny. One thing to note is that we're not definitively saying that it's a new phylum that existed then that no longer exists now. We're just saying that we don't know. So anyway, this is one that's kind of gone between a couple vaguely different definitions. Walcott didn't know. Gould thought new phylum no longer exists. And everything I can find now says that we don't know. So not much about Deinonychus. So go ahead and write down unknown under current interpretation. And lastly, hallucigenia. So Walcott thought this was Annelida, a ringed segmented worm phylum. Gould thought, as he did with quite a few things, that this was an organism that evolved in the Cambrian period and was a phylum then, but that phylum no longer exists today. And you might have in your reading discovered, and you might remember, a critical reevaluation of this fossil that might make it a lot easier for you to identify what it is. For years, scientists were trying to identify this fossil upside down. They thought that these sort of spikes were the legs and that these were like cilia, tactile or sensory organs. And they reevaluated this, and now they interpret it as that these are the legs, and these are spikes coming off of, I guess, the back of this organism. So that changes the way you look at this, and I presented it to you upside down intentionally on purpose and somewhat evilly. But what's our interpretation of this fossil now? So currently we think that Hallucigenia is a very early member of the phylum Onychophora, which is represented by velvet worms of today, and I'll show you what modern velvet worms look like. But it's important to note here again that scientists are not suggesting that this was a new phylum that no longer, then in the Cambrian, that no longer exists today. That was Gould's interpretation. Rather, we're assigning Hallucigenia to a phylum that does exist today. So go ahead and write down Onychophora for Hallucigenia as current interpretation, and then we'll kind of check out what those look like today. Maybe you've seen examples of what velvet worms look like, but I'll show you some images. So these are modern day velvet worms and a variety of species of Onychophora. And you can look at this organism and come to conclusions about some similarities and differences. Obviously the large spikes on the back are absent, but the legs look similar, the head looks similar. And this is what scientists currently think or are currently assigning Hallucigenia to this phylum. One thing you might be thinking at this point, and it's similar to what scientists were thinking when they realized they had been looking at Hallucigenia all wrong, is if we interpret these sort of spike-like appendages not as legs but as coming off of the back of the organism, Hallucigenia, they must be used as a defense mechanism of some kind. And so this led scientists to start searching for a predator class of organisms that was present also in the Cambrian period. And so the story of the discovery of the first sort of Cambrian explosion predator is a really interesting one, and so I'll relate that to you from the fossils. So here are two photos of the same species of fossil, and I want you to take a look at both of these and try to take a guess as to what you think this organism is. So this might agree with what you thought. Most scientists were keying this organism as a jellyfish of some kind, and so they called it Peytoia. And for a long time, that's what this organism was thought to be, present in Burgess Shale type fossil deposits. Here's another fossil from similar beds. So same story. Take a second, look at these fossils, and see if you can decide what type of thing this is. Scientists thought that these fossils looked sort of shrimp-like, so they called them Anomalocaris, sort of anomalous shrimp. And again, for the longest time, that's what these were keyed out as. And in drawings of Cambrian life, you see all this type of stuff. So you can see up there at the top, the person that drew this picture included one of those Peytoia jellyfish looking things, and also included Anomalocaris, weird shrimp thing. And there are tons of different drawings of the Cambrian environment that look similar. Here, in fact, is a zoomed-in front cover of Wonderful Life by Stephen Jay Gould, that we talked about previously, and you'll notice it also has that Peytoia jellyfish and Anomalocaris shrimp on it. But then they discovered this really important fossil, and take a careful look at this, because there are some similarities and differences to what we previously saw. You'll notice that you do see Anomalocaris fossils here. So here's one Anomalocaris, see that there. Here's another Anomalocaris. And you'll notice, though, that strangely in this fossil, they're symmetrically oriented. And they're symmetrically oriented about a fossil that it's difficult to distinguish here, but I mean, the people looking at these have expert eyes. This right here is a Patoia fossil, one of those things that scientists were calling a jellyfish. And weirdly, though, it doesn't stop there, right? This thing is continues with a longer body plan. You can see that the body continues down here and even way down here, looking kind of like that. So yeah, very very weird looking fossil here. And what did scientists make of this? Here's some more accurate drawings that I placed on here showing you where Anomalocaris and Patoia would be on this larger looking organism. And, and as you might have guessed from this evidence, Anomalocaris and Patoia are not separate things. They're not two separate organisms. They're all part of one much larger organism. And this was the first organism discovered to be a predator in the Cambrian. And they still call it Anomalocaris, which means anomalous shrimp, even though this organism is very far from a shrimp. They kept that name for it. So this is a flat fossil. What is this organism thought to have looked like in 3 dimensions? Well, it's a very wild looking thing. And this is a 3 dimensional sort of looking picture that I found online. And on here you can see where the projections that scientists identified as shrimp for a very long time, right here, there's one there, and then there's one on the other side. Projections coming off of the head of the organism. But this organism is very large, orders of magnitude larger than Hallucigenia, Wawaxia, Deinonychus, the things we were looking at earlier as examples of Burgess Shale fossils. But take a second and try to guess where the jellyfish-looking part of this organism is, that thing that scientists keyed out as Patoia jellyfish for a while. Where is that? And the answer is quite startling. So, weird indeed. If you look at this might actually be a model that they made and photographed. I'm not quite sure, but yeah, it's the mouth of the organism. So pretty clearly here you can see the two projections that science for a very long time thought were actual shrimp in the fossil record. There's one, there's the other, and then where's the jellyfish thing? Well, the jellyfish thing, not a jellyfish, is the mouth of this organism. And actually, if you look in the fossil very carefully, there are little sort of teeth in here that are hard. So this organism as a whole is called Anomalocaris, and it is one of the earliest predators that we've discovered. Here's a much cuter drawing of the organism that I found in another textbook to give you a more positive impression of Anomalocaris as opposed to the disgusting stuff that I just showed you previously. But yeah, one of the earliest predators. So let's look at Anomalocaris in comparison to some of the other organisms we were looking at as Burgess Shale fossils. So again, on the left, that's the size of a human being, and you can see Anomalocaris in red is really, really large as compared to, for example, down there at the bottom, you can see drawn some of the organisms that we were looking at in detail. Here's Wawaxia, here's Pikaia, that really early chordate, and then really tiny down here is Hallucigenia. And look how big Anomalocaris is, orders of magnitude larger. And so what this established in the early Cambrian was an arms race of sorts. Organisms that were being preyed upon, evolutionarily adapted over time to develop defense mechanisms like Hallucigenia's spike-like appendages. And predators evolved in response to that. So that made the evolutionary process during this time period even quicker than it already was. So what do scientists currently view as a correct interpretation of the evolutionary tree of life? We've gone through a variety of thoughts about this just during this slideshow. Remember at the beginning we had What Walcott understood, which was sort of a gradual spreading of the evolutionary tree. And then we had a modification of that, which was Gould's interpretation, which was, yes, a dramatic split illustrating the Cambrian explosion. Most of the phyla that evolved during that brief Cambrian explosion period went extinct. Only a small few survived and constituted, as a result, all of the biodiversity that we see today. And the main factor that Gould suggested was chance. So no one really believes that anymore. Remember that scientists have been able to connect most of the Burgess Shale fossils to currently existing phyla. So this is a look of a more current interpretation of the evolutionary tree of life. Some things to draw your attention to. One, this is definitely not what Walcott's definition of it was, because it includes, critically, the existence of a Cambrian explosion. That after billions and billions of years of relatively stagnant, slow evolution, little squishy life not doing much, 10 million year period starting around 550 million years ago, we had a rapid evolution, rapid diversification of almost all of the known phyla of organisms. And henceforth, yes, we've had more diversification, but nothing like what we saw in the Cambrian explosion. And you can see that, yes, some of those phyla went extinct, for sure. But the thought is now that that is not a commonality. Rather, it's kind of an exception to the norm, and most of the phyla that evolved in the Cambrian explosion can be still traced to currently existing phyla. So here on one slide, you can see really over the past century, maybe a little bit more, the view of evolution has changed radically from a very traditional interpretation like the one of Walcott and like the one of Darwin to really wild controversial interpretations like the one of Gould to our most accepted current interpretation, the one at the bottom. So it's important to know that science is always changing and always advancing, and what we deem today as being a correct interpretation might, just in a few years be turned completely on its head. You can pause it here if you want and take a look at this really, really crazy graphic that I pulled from the internet that I think was a really neat illustration of geologic time. Kind of shows in radial arcs the origins of life and expanse diversification of life to the current diversity of life as shown in this big, huge arch right here. But you can see the Cambrian explosion, and you can see how much of the diversity of life evolved there. You can also see mass extinctions, which are interesting to note on here. So if you want to pause it and take a look at this chart, I think it's pretty neat. You can get a lot of information from it. So I'd like to take a moment here at the end of this presentation to tie in some of the goals of our course, which is really to illustrate how the physical world yields changes in the biological world and vice versa. So this is a review of a lot of the information that was brought up in the When Life Exploded article from Time magazine. So again, you're definitely going to want to read that if you haven't done so already. But we can summarize hypothesized triggers for the Cambrian explosion into four categories: oxygen level rising, Hox genes, predation and arms race, and what the article calls the empty barrel theory or hypothesis. And these are all providing reasons for why, strangely, after billions and billions of years of not much happening evolutionarily and in terms of diversification of life, we have a very short geologically time period where stuff randomly starts happening at a very, very quick pace. So why did it happen when it did? Why did it happen so quickly? That's what these hypothesized triggers attempt to explain. So one of the main thoughts behind why the Cambrian explosion happened when it did is that oxygen levels on Earth rose dramatically near the beginning of the Cambrian. And to understand why that is, let's first talk about what was going on during the Precambrian, and we'll elaborate on that more when we get further back in time later in this course. But it's important to know that the first organisms that are photosynthesizing appear around 3.5 billion years ago. And remember that life's origins date to around 3.8 billion years ago to give you some comparison. And photosynthesizers are consuming carbon dioxide from the atmosphere and producing oxygen. And that led to a definite increase in atmospheric oxygen, but nowhere near the amount that is currently present in our atmosphere. The main reason why it didn't lead to a huge increase in the amount of oxygen is that the oxygen that was produced by photosynthesizers was being consumed by aerobic microbes. Basically, organisms die, and aerobic microbes use oxygen to metabolize dead stuff and produce carbon dioxide. So, what was the carbon dioxide that was being consumed and oxygen that was being produced by photosynthesizers is counterbalanced by oxygen being consumed and carbon dioxide being produced by aerobic microbes. So oxygen levels definitely rose during this period, and evidence of oxygen is present in the rock record as rust, but nowhere near to the levels that we see in the atmosphere today. So, to summarize, there's no dramatic increase in oxygen throughout the entire Precambrian, only subtle increases due to the evolutionary origins of photosynthesis. What happens in the early Cambrian period is that oxygen levels dramatically rise, and they rise to levels almost near what we have today. And there are a variety of reasons for this, and these are reviewed in the article When Life Exploded. But the geologic record suggests that immediately before the onset of the Cambrian period, around 600 million years ago, there was a ton of mountain building and tectonic activity. And that resulted in a lot of erosion, primarily because land plants hadn't evolved at this point. And as you probably know in this class, or from this class at this point, land plants are very good at erosion prevention. The roots get down into the soil and prevent erosion. So when you have a lot of tectonic activity happening, well, that's just soil shedding off into the water, tons and tons of erosion. And so the thought is here that a lot of the fuel for aerobic microbes, dead organic material, dead photosynthesizers and other things, gets buried beneath this sediment that is shedding off of continental margins due to all of this mountain building. And because it was buried beneath sediment, it's no longer accessible to those aerobic microbes. So basically, we're taking away one piece of that counterbalancing puzzle. Photosynthesizers are still consuming carbon dioxide and producing oxygen, but aerobic microbes that were counterbalancing that process throughout the majority of the Precambrian aren't allowed to do so as well because all of their energy source, all of their food is being buried underneath layers and layers and layers of soil because of all the mountain building. So, photosynthesizers still working great, aerobic microbes not working as well. The result is a dramatic increase in atmospheric oxygen. So that's one of the reasons why scientists think that oxygen suddenly rose during the early Cambrian. And it's important to note, obviously, I love these physical reasons because the goals of this class, but this is specifically a geologic process that resulted in an atmospheric change. It's a geologic process that impacted biology, that resulted in a global change, and one that affected biology so intensely that it resulted in an explosion of life, a diversification of life at a very, very rapid rate. And so you might be wondering, well, what's the deal with oxygen? Why is oxygen needed for this crazy Cambrian explosion of life to happen? So for organisms that live off of oxygen, oxygen is the major energy source, and that proceeds through the mitochondria, as you might remember from biology class. And to get more complex organisms, multicellular organisms, organisms that have enhanced communication, signaling between cells, you need more energy. And so this dramatic uptick in oxygen allowed for organisms to have more of that energy to evolve further, which allowed for the evolution of larger things, and as well the evolution of hard parts, teeth, spikes, bones that we see in the fossils of the Burgess Shale and other similar beds. And so what the thought is this dramatic rise in oxygen allowed for more evolution, and what quickly happened is that there was a change in the amount of what are called Hox genes that organisms have. And Hox genes, or homeobox genes, are genes that control the look of the organism along its body axis. So if you look at a developing embryo and how it starts forming different appendages coming off of it, a head coming off of it, legs, things like that, all of that is controlled by Hox genes. They determine the different types of appendages, the vertebra that form along the body plan, things like that. And mutations of specific Hox genes result in dramatic phenotypic variation in organisms. A great example of this is Drosophila. So Drosophila, melanogaster, the fruit fly, which you probably remember also from biology class, lots of experiments done on this organism. We know its genotype very, very well. Has 8 Hox genes, and you can see those 8 Hox genes. They're present on the chromosome in this order, so I'll try to circle one. There's 1, there's 2, there's 3, and so on. And there's a really neat collinearity between the order of these Hox genes on the chromosome and the actual body plan of a Drosophila. And they've done studies where, for instance, they've gone in and inserted mutations in this particular ABDB Hox gene. And what that results in is mutations in specifically this part of the organism right here, the tail end of the abdomen. They've gone in and mutated this DFD Hox gene on Drosophila, and that results in mutations in this part of the organism's head. And you can see that the order is exactly the same. So as you proceed down the chromosome and look at the Hox genes, you're proceeding down the body plan of the organism. It's really neat. Now this is almost a unique example in a sense, because not all Hox genes work like this. It's not like humans have all of our Hox genes on a single chromosome and you go in and edit, and I'm editing the head of the human being, and then I go down and I am editing the tip of the foot. No, not at all. But I think this model is pretty neat because of its simplicity. And it definitely gives you a good idea of what Hox genes are, genes that control the body plan of an embryo. So you might have already guessed, since we started at a simple example of the fruit fly, that if an organism has more Hox genes that control its body plan, then there's more variety for what that body plan can look like. So flatworms have 4 Hox genes. Flatworms look very similar to each other. Fruit flies have 8 Hox genes. Primitive chordates have 10. Humans have a whopping 39 Hox genes. So yeah, there's a ton of different things that can happen to the developing human embryo that are all controlled by one or more of those 39 Hox genes. The evolutionary thought for what happened in the early Cambrian period is that Hox genes passed a critical number, allowing for an exponentially greater amount of diversity of life. And also a dramatic increase accordingly in the speed by which evolution is taking place. And this is a review of stuff that was mentioned in the article that you read. But it's important to know that scientists are almost certain that Hox gene development was involved in the early Cambrian in terms of speeding up the rate of this evolutionary process. And the reading also mentions that with more oxygen in the atmosphere, organisms have more energy, maybe with passing a critical threshold of Hox genes. The development of the gut during this time period also reinforced this process, because what you can think about is prior to the gut, organisms were just exuding waste material out of themselves. There wasn't any compacted form of waste material prior to the gut. The evolution of the gut, you now have fecal pellets of organisms, and the article actually uses this fecal fecal pellets being express mailed to lower ocean depths. If you have something that's a concentrated amount of waste product, well, it descends to ocean depths where it's no longer accessible to aerobic microbes. Remember, aerobic microbes are using waste material and things like that as their energy source, through which, when they metabolize that energy source, they're consuming oxygen from the environment. So, if fecal pellets exist, again, aerobic microbes have less food, and they're less able to counterbalance what photosynthesizers are doing to increase the oxygen in the atmosphere. So this is another thing that scientists think led to even more oxygen in the atmosphere and an even greater speed to this Cambrian explosion of life. The article also mentions, and I've alluded to it before, the development of predation in the Cambrian period, and we saw the history of the discovery of Anomalocaris, early predator. The evolution of a predatory class results in evolutionary speed because then there's an extra element of selection happening. So an arms race of sorts develops between predator and prey. An example being Hallucigenia versus Anomalocaris. And the article also mentions something that's called the empty barrel hypothesis. The thought here is that, well, yeah, if organisms are developing at a rapid rate to be able to do different things, well, then they're able to access niches that were previously empty. The article strangely compares it to a bunch of empty barrels that need to be filled. Basically, what they mean by that are vacant ecological niches that organisms evolve enough to where they can expand into those niches, and that dramatically increases the rate of evolution as well. So I think it's cool to summarize these types of physical and biological tie-ins for why the Cambrian explosion happened when it did after billions of years of rather stagnant biological activity. So that's the Cambrian explosion, a really unique event that happened only once in the history of life on this planet, around 550 million years ago, a very brief 10 million year period where life was evolving very, very quickly. And if you recall, almost all of the phyla that we have on Earth today evolved during this very brief 10 million year period. It's pretty amazing to think about.